Hi, everyone. Thanks so much for joining us at Venture Cafe tonight. So my name is Angela McQuillan. I'm the curator at the Esther Klein Gallery at the Science Center in Philadelphia, which is also home to Venture Cafe. Tonight, I'm very excited to present the work of artist Laura Splan, who has presented her work at Venture Cafe and the Science Center many times before, but today we're celebrating the opening of her exhibition titled Disrupted Domains that just opened in the drop-in lounge of Quorum at the Science Center. So Laura Splan has done 2 Bioart residencies with the Science Center inside the laboratories of Integral Molecular. During her first residency, she spent 3 months working in the lab alongside scientists, and her research culminated in a solo exhibition at EKG called Confirmations in 2019. And then this year, Laura came back for a second residency, this time a virtual collaboration where she built upon the ideas in her exhibition to create a new series of animations that are part of a project called Precarious Structures. So I'm really excited for her to talk more about this whole process, and it's always such a pleasure to work with Laura. So a brief bio, Laura Splan is a Brooklyn-based artist whose transdisciplinary work intersects science, technology, design, and culture. Her biomedical themed artworks have been commissioned by many institutions, including the Center for Disease Control. She has exhibited her work nationally and internationally, and her work has been featured in prominent publications such as the New York Times. Laura's work has been supported by many grants, including the Jerome Foundation and the Pollock Krasner Foundation. Laura is also a creative science member at New Inc., the New Museum's cultural incubator in New York City. Her latest solo exhibition, Unraveling, was featured at Bioart Art Space in the Brooklyn Army Terminal. So now I'd like to go ahead and welcome our guest, Laura. How are you?
Good. Thank you so much, Angela. Thanks for having me back.
Of course, as always.
Yeah, it's nice to be able to present the new work that I'll be talking about and also talk about some older projects that have led to it and also how the work kind of keeps evolving and coming back to materials and ideas that were started before and during my first residency in 2018. So I'm going to share my screen. All right. Well, yeah, it's really great to be able to talk about this work and some of the projects that led up to this exhibition of these new animations in Quorum at the Science Center. And I'll be talking about the animations that are on view on Market Street, as well as previous projects that provide some context to how I came to create them. And my work as a whole explores intersections of science, technology, and culture. And I make conceptually based artworks that connect the material artifacts of biotechnology to reveal poetic subjectivities of the seemingly mundane. And I try to destabilize and reframe our relationship with the everyday, and to even redefine what the everyday means. And I created this lace SARS doily in response to the first SARS outbreak in 2003 and how it kind of provoked an increasing awareness and unease with the microbial world. And this project was grounded in the intricacies of textiles and its associations with the familiarity and comforts of domesticity, while also being driven by a deep fascination with the seductive and sometimes alien representations of the biological world. And I often try to compel an intimate engagement with detail, calling into question how things are made and what they are made of. And I use processes and tools that challenge notions of what is made by hand, what is made by machine, what is natural and what is constructed, and what is science and what is culture. And my series of doilies leveraged the formal conventions of lace by superimposing the morphology of enveloped viruses onto the traditional radial doily forms. And today, after exhibiting pretty widely the series for almost 2 decades, the doilies themselves have inserted themselves like a virus into the collective consciousness that is Google image search, when searching for both doilies and for viruses. And the project led me deeper into the layers of scientific representation, biological imaging, and the ways that bodies are translated and abstracted with technology. And I'm especially interested in the history of inventions of scientific apparatus and the cultural circumstances in which they are conceived. And the history of René Laennec's invention of the stethoscope in 1816 is fraught with varying accounts of its conception that cite gender relations as much as technical research and auscultation. One version of its invention tells a story of a quote unquote well-endowed female patient who by some accounts was too modest to have Laennec lay his head on her chest. And by other accounts, it was he that was too modest. Whatever the reason, Laennec's solution to this socially awkward moment was to roll up a sheet of paper to create his first iteration of the stethoscope. So the stethoscope itself embodies a social paradigm, a prescribed comfortable distance between doctor and patient, male and female. And in my stethoscope sculpture created in 2002, I extended the length of the instrument to 25 feet, the longest distance at which one can still hear a heart beating at the other end. And its absurdity belies its continued, albeit diminished, function and questions the technical standards on which an institutional device is based. And my embodied object series uses electromyography sensors to create data-driven artworks using this common medical diagnostic technique. So here, neuromuscular activities associated with experiences of wonder were performed as facial expressions and bodily movements, such as smiling in delight, blinking in disbelief, and frowning in confusion. And each activity produced unique data captured by an EMG sensor that was translated into a curve using custom software, which I then used to create data-driven patterns and forms for 3D printed sculptures, such as smiling on the left and blinking twice, pictured on the right. And I also created data-driven patterns for computerized jacquard weavings. With this tapestry titled Undo, I recorded muscle movement as I was unraveling another weaving. And I often seek out these recursive and hybrid combinations of materials and processes with technology to challenge values of the hand in creative production and question notions of agency and chance in aesthetics. And so this fringe effect on the perimeter of the weavings was created by unraveling individual threads by hand along the machine woven edges. And this act of unraveling was particularly evocative and murky territory to me. What does it mean to unravel the work of a machine? Is the work of a machine less valuable or precious than that of the hand? And with this process, as with other projects that I engage my fingers beyond the operation of a mouse, I often find myself thinking the word undo in my head when making a mistake in some sort of moment of absurd confusion of corporeal and technological experience, existence, and agency. And sometimes this word, which I seem to be trying to select from some imaginary sideward drop-down menu, is even replaced by its keyboard shortcut, Command-Z. And then there's the realization that I'm not working in a digital space furnished with history palettes and autosaves, but rather a meat space equipped only with my precarious and clumsy body. And these paradoxical relationships between the body and technology can reveal something about the value we place on the labor of each. And in this series of performance sculptures entitled Material Expressions, EMG electrodes attached to my chest actuate a motor that unravels a scarf that I'm knitting. And my task as performer and as body is to remain calm, to slow down my heartbeat, to slow down the motor, to knit faster than the motor can unravel my knitting, or at least to maintain a state of equilibrium. And these sensor and data-driven projects are part of a larger investigation of the body as material, as interface, and as data. And in 2018, I was invited to be a bioartist in residence at the uCity Science Center in Philadelphia, where I was hosted by biotech company Integral Molecular, whose research includes antibody discovery. And I attended lab meetings where I took copious and cryptic notes. And I also had unique access to move around the lab, taking many photographs and videos of often unseen vignettes of biotechnology. And I was particularly drawn to the lab machines with their quirky interfaces and their often absurd language that seems now more than ever so oblivious to the problems of the outside world of the living. And the images served as a document of the numerous biotechnological artifacts I observed in the lab. And I also compiled them into a book of photos with accompanying texts that I'll be sharing as a PDF during the Q&A. And this particular spread has a conversation with an AI chatbot that starts with me asking if they knew anything about molecular biology, which is answered with, there are a lot of them, real and fictional. Point being, I'm unique and yours. So you can read the rest of this conversation and exchange, as well as the other text and see the other images in the PDF that I'll share the link with in a bit. And I also had the opportunity during this residency to experiment with some of the software that the scientists were using for their antibody research. And this molecular visualization software called PyMol works with models that are publicly available in online databases. So of course, the first thing I found myself doing was unraveling protein structures, such as this model of a ricin toxin bound to a camelid nanobody, or structure 4Z9K in the protein databank. The camelid referred to was specifically an alpaca. Alpacas and llamas have unique antibody structures called nanobodies that are easier to work with in the laboratory. And they have many advantages over antibodies in terms of stability and cost. And I was struck by the lab's use of non-human species for the production of antibodies for vaccines and antiviral treatments. And my conversations with the scientists led to receiving over 200 pounds of wool from laboratory llamas and alpacas who produce these nanobodies for human drugs, including vaccines and antiviral treatments. And since then, I've been hand washing, carding, and spinning their wool into yarn. And in my exhibition in 2019 at the Esther Klein Gallery last year, I situated sculptures made with this hand-spun yarn among other artworks that examined the hidden systems and invisible labor of biotechnology. And this work questions notions of the presence and absence of bodies, evoking immutability of categories that delineate their status. And Lumen choreographs viewers' movements to sit on a rug made with the wool of these laboratory animals. In biology, the lumen is the interior part of a cell where a protein is folded and modified. And so here, sitting on the rug engages viewers with these unseen materialities and labor as they touch the yarn and listen to an accompanying soundscape that layers recordings I made in the lab during my residency. And another component to this project and research is an interrogation of the language that's used to explain science. This is another piece from my EKG exhibition that included appropriated text from the book Molecular and Cell Biology for Dummies. And the book equates plasma, or this excerpt in the book equates plasma membranes with international boundaries and cast proteins as customs officers. And the antagonistic implications of this metaphor were especially heightened by the Trump administration's zero-tolerance immigration policies in 2018, for which we continue to experience the results of. The gallery behind the curtain invited viewers to sit on a bale of hay to watch a molecular animation made with a model of an alpaca nanobody in complex with the ricin toxin. And by using the specialized features of the software in unconventional ways, I created animations that are collaborative doodles of sorts between the software, my hand, and the molecular structures that are being disrupted. So as the protein structures are manipulated, the software renders uncanny disturbances in the form of sometimes spastic and sometimes sublime movements. And these videos are unedited screen recordings of performed interactions where my mouse is hidden. So the labor of the artist and software is collapsed by the seemingly autonomous moving form. And while I was processing the llama wool in my studio, I would often find clumps of feces, which I began to collect for this networked laboratory mixer that is agitated by Twitter. And the device activates when Twitter hashtags associated with the culturally contested status of science are tweeted. So here, the mere mention of hashtag global warming or hashtag vaccination agitates tubes filled with laboratory animal feces. Since taking office, the Trump administration has advised how to improve the chances of receiving research funding with the suggestion to avoid words and phrases like vulnerable, diversity, entitlement, transgender, fetus, evidence-based, and science-based. And the administration has also refused to sign statements that mention climate change. As visitors exited the exhibition, faint text on a wall invited them to come close to read, "Our distance allows our intimacy." And the phrase refers to the complexities of existence in the biotechnological age, where understanding of our own bodies and the bodies of others is increasingly mediated by technology. And the sculpture blows a breeze in the viewer's face as they read the text. And the speed of the networked fan intermittently adjusts based on the wind conditions near a farm in Western Pennsylvania. And this farm is actually the 600-acre biological laboratory that gave me the wool from their 2018 shearing of their llamas. And in January this year, they invited me for a 3-hour tour of their sprawling facilities to meet and photograph the llamas and alpacas, as well as photograph the laboratory where they process their biological products, such as blood, tissues, and organs. And I'm hoping to return to the farm post-COVID to resume working on photo and video-based work. Shortly after my visit to the farm, I did a Bioart residency at Coalesce at the University of Buffalo. And I wanted to see what would happen if I brought this llama yarn back into the laboratory. So I came to Coalesce with the goal of genetically manipulating bacteria to produce pigment to be used to dye this yarn. And to do this, I used a lab technique called Bacterial Transformation. And it's a process where a cell is genetically manipulated to incorporate DNA from its surroundings through its cell membrane. And this is a commonly used lab technique that's used to introduce a foreign plasmid or DNA into bacteria. And then the bacteria amplifies the plasmid, making large quantities of it. And a plasmid is just a small circular piece of DNA that contains genetic information for the growth of bacteria. And I was working with a few different plasmids that might be used to dye the yarn in combination with E. coli bacteria, which is often referred to as a workhorse of the laboratory. And for transformation to take place, the bacteria must be in a state of competence, having small holes in the bacterial cells for a DNA plasmid to pass through. And I was particularly interested in the narrative implications of this transgression of boundaries as it was being choreographed in this decidedly Baroque approach to artificially producing pigments that already occur naturally in other bacteria. And the residency supported me in the development of several protocols I would need in order to transform bacteria to produce different pigment and color for dyeing the yarn, including fluorescence, seen here with a black light or UV light, and purple, blue, and also green. And we tried a number of different approaches, including soaking the yarn in bacterial cultures, and also experimented with producing large quantities and different colors, including the green and purple and fluorescent, and also comparing undyed and dyed fibers of yarn. And soon after my return home, after a month in Buffalo, when I returned home to New York City went on lockdown for the COVID-19 pandemic. But during this residency, I did have the opportunity to teach a bacterial transformation workshop, during which we also discussed the language used to explain cellular biology. And my goals of the workshop were to make the science accessible and demystify laboratory protocols and tools, but to also think critically about language used to explain science. And the workshop included a discussion examining metaphors and meta-narratives in biology texts, while brainstorming for new language and metaphors to explain the science of bacterial transformation. And some of the texts that we were examining, again, from the molecular biology for dummies for cell and cell biology as well. Some of the excerpts that we were examining are fraught with antagonistic metaphors that are grounded in narratives of trickery. So here a virus tricks the cell into bringing it inside, or competition. Bigger molecules will be closer to the starting line, smaller will be closer to the finish line. And surveillance, defensive proteins wander by to make sure that everything is okay. And assimilation. Chaperone proteins assist folding of new proteins and guide proteins to their proper locations within the cell. And when I returned home to New York City, it went on pause with shelter-in-place policies for COVID-19, but I continued to collect more metaphors and language emerging out of the communication of science around the coronavirus, finding numerous sources for inspiration in the media, including this April 3rd New York Times article called "Bad News Wrapped in Protein." And it's populated with a motley crew of the usual suspects like saboteurs and tricksters, and eventually the end of the line, which I'll actually come back to. And I also returned, while sheltering in place, to my molecular software animation experiments that I began in 2018 that I could work on my computer at home. And I began collaborating remotely with scientists at Integral Molecular, where I did my 2018 residency for the production of new molecular animations. And the new animations continue to explore the unraveling and distortion of the folded proteins in viruses and antibodies. And this video, this is an excerpt from a rather lengthy, again, kind of software performance video called Denatured, and it's made with a SARS-CoV-2 spike protein receptor binding domain in complex with a llama nanobody. So over the duration of this piece, I slowly unravel both structures. And the remote residency also consisted of Zoom meetings with scientists at Integral, including CEO and virologist Ben Durans, and communication scientist Edgar Davidson, to discuss their research on SARS-CoV-2. And our meetings wandered into discussions of subjectivity and molecular visualizations, as well as the scientific and creative implications of the software's features. And as I shared animation tests, new conversations emerged around such topics as the aesthetics of the software and the role of the spike protein structural changes in infectivity. And these animations are part of a continued exploration of interconnected systems as they are affected by both human and natural forces at macro and micro scales. And my current work with the software is interested in evoking the precarity of underlying structures that have been rendered so visible during the coronavirus pandemic. And I've been spending the last several months studying the new features and tools in PyMol, including the ability to automatically animate or morph two different conformations or structural forms of a protein structure. So a conformation is just different forms of the structure, such as the unfolded or folded structure. And I've also been experimenting with the morphed animation of the spike protein conformations in particular. And I've been using the morphing feature to animate SARS-CoV-2 structures that I've unraveled by hand and experimenting with speed as well. So the software itself does have not only the ability for me to unravel the proteins, but also to create the animations of that folding and unfolding within the software. And I'm able to export the animations to be used in more conventional video software for further processing, such as duplication and rotation. And I've also been obsessing over not only the palette of colors available in PyMol, but the names of the colors as well. The colors in the software's palette include references to nature, such as plants, animals, and chemical elements, and references that are often entangled with idyllic representations of the natural world, such as blue skies, green forests, fruit, and flowers. And they present additional layers of abstraction built into the interfaces of the technologies with which we engage with and manipulate the natural world. And they function to provide both simultaneous simplification and complication of form and understanding as it becomes increasingly difficult to define what is nature and what is natural. And I was doing a lot of experiments with this lemon color, this really bright, vibrant green. And so I kind of went down a rabbit hole of lemon and what is lemon or lemon and what are the, what is its genetic lineage. And I was also kind of interested in this increasing distance we have with the natural world through our everyday engagement with even the most basic needs, like food, that become increasingly abstract and convoluted. These different colors upon colors and nature upon nurture. And even looking at these kind of ridiculous reviews of this product that says Cheetos crunchy flaming hot snacks can literally take your breath away. So that lemon was a bit of an obsession for a second, and I started to use that color on some of these animations and very selectively adding color back into what I was starting with as colorless or white and gray forms. And as I continue to work with the animations throughout the summer, I eventually started to kind of play more with this kaleidoscopic effect as well. And so eventually the series kind of landed on this mesmerizing meditation of these increasing entanglements between natural and constructed worlds that are mediated by technology. And so I was starting to play quite a bit with the unraveling of the proteins in the software and animating that within the software in terms of folding and unfolding and also orbiting around the 3D structure of the model. And then processing those into these continually and increasingly mesmerizing animations, where the palettes for each are specifically chosen for their name. So this particular animation is called Lime, Limon, and Forest, and uses those colors from the Pymol color palette. And this one is called Marine, Aquamarine, Sky Blue. And so I think I have one more. This one is Orange, Oxygen, and Salmon. And I was really enjoying playing with these kind of precarious and delicate and often kind of sublime movements of the animated folding and unfolding of the proteins. And just as I was finishing those animations, sometime in the summer, I was invited to do a residency at Bioart space at the Brooklyn Army Terminal. And that kicked off with my current solo exhibition that features these animations projected in their 15,000 square foot dark space gallery, along with an immersive soundscape, which I'll speak about in a bit. So this is an installation view of the animations projected in the space. And the gallery, Bioart Space has two galleries. This is the dark space gallery. And there's also a front main space gallery that is on the Brooklyn Army Terminal waterfront. And after New York began to open back up in June, I was able to do a small timed viewing of the exhibition. And so I also, for this exhibition, began to expand my use of the wool and to connect it with the unique experiences and traumas of this very curious pandemic moment. And one of the projects I'm developing during the residency includes recordings of Zoom sessions with both friends and strangers around the world. And during these sessions, we unravel textiles while discussing the pandemic in wandering conversations that range from the personal to the political to the social. And so the project is part oral history, part conceptual craft, part performance art. And the project attempts to unravel our unique and shared experiences of precarity, trauma, and resilience during the COVID-19 health crisis. And participants are mailing me their unraveled threads from these sessions, which I'm then weaving into tapestries that incorporate the wool of the laboratory animals. So on this previous slide, you can see the skeins or balls of yarn that I've spun from the laboratory llama and alpaca wool. And I like using these textiles, materialities, and gestures in both real and virtual spaces to examine our entanglement with the biological world and cultural constructions by which we engaged with it, and constructions of worlds that are so often mediated by technology. And while I was spinning and unraveling threads and virtually denaturing proteins, I couldn't get the echoing melancholy of the repeated adenine nucleotides in the SARS-CoV-2 mRNA sequence that was depicted in that New York Times article. You can see here at the end of the sequence, there are 33 A's. And so I started to invite biotech scientists and workers to play the sequence over Zoom on a guitar. And the series of recordings was made by prompting them to play A on guitar 33 times, the number of adenine nucleotides at the end of the sequence, which is called a termination sequence. And I'm going to switch my audio here. So that live acoustic guitar performance was by integral molecular biochemist and co-founder Joseph Rucker. And it became the backdrop for this installation at the Bioart. And the slow and persistent plucking of the notes, I sort of loved for its ability to be at once soothing and stressful and invisible and present. And I also Zoomed with lab instrumentation engineer Frank Masciocchi, who is also a musician. And Frank's electric guitar had an entrancing effect as he riffed on the termination sequence with an array of pedal effects and loops in what was nearly a 20-minute performance that was aptly framed from a loss of light as the sun set. And I quickly realized that the fuzzy and goopy interpretations that he was doing of this prolonged string of A's was really destined to be the soundscape for the installation at the Army terminal. These animations are currently on view through December 18th in Quorum at the uCity Science Center, and visitors can scan the QR code posted in the lounge to listen to the soundscape on their own smartphone as they encounter the screens mounted throughout Quorum. And I'll share a few links in the chat in a moment that will give you some more information about this and other exhibitions. But now we have a very special treat of listening to Frank perform from one of Integral Molecular's lab. So I invite everyone to sit back and enjoy this brief performance, followed by the Q&A, where we'll also be sharing a link to download the artist book I mentioned that I created during my first Science Center residency. So I'm going to stop sharing my screen and then
Frank here.
Frank can get started.
Great. Thank you so much, Laura. That was a beautiful talk.
Yeah, oh, thank you. Thank you. I'm really excited that Frank is able to perform and from the lab to boot. So Frank, whenever you're ready, you can give us a thumbs up and take your time getting into position. He's got to switch his headphones and technology.
Cool.
And I will load up some links in the chat too while that's happening.
So one quick question.
Sure.
Did you create all these animations just in PyMol, or did you use other programs as well?
Yeah, so I do the folding and unfolding and the orbiting on the model in PyMol, and then the animation of that in PyMol. And then I'm able to, what I've been doing is exporting or animating and exporting a single protein structure.
Mm-hmm.
And I'm able to export that at really any resolution, which is kind of amazing. I'm really only limited by my computer. And then bringing that into other video software, I've been using After Effects to create the kaleidoscopic repetition. But everything else is controlled in PyMol, the color and the speed and all that.
It's really impressive because, you know, my first experience with PyMol was that like fluorescent green color, and you made these like gorgeous color combinations. So
thank you. Yeah, it was an interesting set of parameters to limit myself to, the nature palette.
Yeah, definitely.
So Frank, do you want to get started?
Thank you so much. That was awesome.
I'm going to share the links in the chat too.
Great.
So the first link is more information about the exhibition at Quorum, and has the full sound file that you can play of the Zoom performance that was recorded and is now I see as kind of permanently attached to these videos. So I'm really glad that people will be able to listen to them on their smartphone in Quorum. And then I also included the PDF file if you want to download the Needle in a Haystack book.
Thank you so much.
Yeah.
I just want to go back to, we have a couple questions in the chat. I want to go back to Tracy's question. I know there's a couple people from Integral here, including Ben Durant's. Does this cause you guys to look at the molecules or your research differently? And has it caused any breakthroughs?
Well, I'm not sure if that's directed to me, but great performance, Frank and Laura. Wonderful talk. You know, artists here make us look at everything differently, and we've actually had that question before, and a number of scientists have told me that Laura and Deirdre and Heather, a number of our artists have really changed the way that they think about certain experiments. Certainly, Laura's experimentation with PyMol and enabling it to have different features we've never done before, but really is inspires us to think of protein structures differently and how to manipulate them. So it's always been an inspiration, and certainly what Laura's done with SARS, the structures, and taking something that's, obviously affecting all of us and making it something beautiful in an amazing way. So I hope that answers some of the question.
Yes, definitely. Thank you, Ben. It's great to hear from you. We have like 2 minutes left. Does anyone have any other questions for Laura?
I just want to quickly point out that it's probably a safe assumption to make about looking at Ben's situation right now with the 3 guitars on the back of the wall, that there was some influence there in my Zoom meetings with Ben during this collaboration, that somehow I thought, guitar might be a good idea here.
Subliminal messaging.
We have here a comment from Ruben. Your work was already exploring intimacy and distance through technology, biology, interactivity, and collaboration. It's so wild how the COVID pandemic forced this even more in your way of working. Can you speak to this at all and how you had to adjust your studio practice or refocus projects?
Yeah, I mean, one thing that I realized very quickly, even around the previous work, just even talking about previous work, much less conceiving of new work, was how all of a sudden I didn't have to explain a lot of things that I previously had to explain. I didn't have to explain what SARS was and what its significance was. And so there's sort of topics that I've been dealing with that used to be more niche or kind of I think difficult to understand. All of a sudden there's a certain common language that more people have in relation to not only accessing the new work, but also looking at the previous work like the doilies. And I think that in many ways that is liberating, that there's a certain new way of navigating the creation and the discussion of the work. But yes, the pandemic certainly presents new challenges, I think, in considering the sensitivity of the subject matter. and the kind of multi-layered quality of it.
Definitely. Well, thank you so much, Laura. We're out of time, but this was amazing. And thank you so much, Frank. Very informative talk. And if you want to see Disrupted Domains, Quorum is open every day, Monday through Friday from 10 to 3. Walk in anytime. And thank you, everyone, for joining us.